Cell respiration is a multi-step metabolic pathway where we take organic molecules like glucose, fatty acids, amino acids, and break them down in a very controlled way by a series of enzymes. During this breakdown process, potential energy is stored in the molecules' chemical bonds. That energy is now released. The energy is used to make new bonds that change ADP, antifree phosphate, to ATP. So these processes are exergonic or energy releasing. The organic molecule that gives up its energy is done by releasing high energy electrons. So that molecule is oxidized. The energy released is going to be used to synthesize or make ATP, which is endergonic or an energy requiring process. Remember, it requires energy to build. So oxygen is required for maximum ATP production. And even though there are different types of organic molecules that we can chemically digest during this whole process of making ATP, cell respiration, that's making ATP, we're going to focus on the oxidation of glucose. So I want to overview this with you on the diagram that you have on the right side of your screen. So here in the pink box, we start with a molecule of glucose in the cytoplasm of the cell. It undergoes the first step of cellular respiration, which is glycolysis. Now look at that word, glycolysis, sugar splitting. In the process of doing that, we gain 2 ATPs. Glucose gets converted to something called pyruvate or pyruvic acid. If there's not enough oxygen present, so say you've been working out for a long time, then pyruvate is going to undergo fermentation to build up lactic acid, which causes muscle fatigue, or ethanol, which is alcohol, like in your wine. Now, when there is plenty of oxygen present, that pyruvate is going to undergo something we call transition, which is sort of like an intermediate step between glycolysis and the Krebs cycle. Now remember, glycolysis occurs in the cytoplasm of the cell. The Krebs cycle, also called the citric acid cycle, the TCA or tricarboxylic acid cycle, they're all the same thing. I don't care which one you call it, okay? Just know what you're talking about. So this is also in the cell, but it's within a specialized organelle called a mitochondria, okay? So in the Krebs cycle, we continue the breakdown of glucose. We create some waste product called CO2. We create a tiny little bit of ATP. And then we're going to move to a different part of the mitochondria, to the inner layer where we have something called the electron transport system. And when there's plenty of oxygen available, man, we start pumping out ATP like crazy right here in this last step. So let's review that again. The first step is glycolysis. It happens in the cytoplasm. We make a little bit of ATP. Then if there's plenty of oxygen present, we have transition of pyruvate to something else we haven't named yet, that then enters the Krebs cycle inside the mitochondria that's in the cell. Now, during that process, we make some waste product, CO2, we make some ATP, and then we take all of the stuff that we put in our carrier molecules, that NAD and something else we'll talk about in just a second. We take it to the electron transport chain and we use that and oxygen to make a whole bunch of ATP and some water. Now guys, that seems complicated, but I can promise you that's the easy version, and we're going to keep it pretty simple. It can get pretty darn complicated. So this is just something a student drew out, and I can promise you this is still not all of it. Okay? There's a whole lot to this we're going to get to the meat of it so you understand what cell respiration is all about. The energy transfer from chemical bonds in glucose can be used either directly, which is the least common way, or indirectly, which is the most common way, to make that ATP. The direct method of making ATP is called substrate phosphorylation. The indirect method is where the energy is first released to coenzymes, that NAD and FAD. Then they transfer that energy to form ATP. That is a much more complicated pathway. We call it oxidative phosphorylation, and it involves at least 20 different enzymes. So let's take a quick look at this. Here we have substrate level phosphorylation. Now remember, phosphorylation simply means to add a phosphate. Well, what are we adding it to? We're adding it to the substrate, okay? So here we've got a phosphate group. The purple is our enzyme. The yellow here is ADP. Now tell me again what ADP is. Adenosine diphosphate, which means it only has 2 phosphates. We can't use that for energy. So what do? The enzyme binds to the phosphate group, binds to the ADP, and transfers this phosphate right here onto that ADP. See, it's missing there. And it's right here. We now have ATP, adenosine triphosphate, because those 2 added to that 1 gives us ATP. Okay? Now, in the oxidative phosphorylation, we have to go through all those steps. Okay? We've got to go through glycolysis, the transition, the Krebs cycle, and the last part is this electron transport chain where we use oxygen and those electrons to make ATP and water. All right, just a refresher here. The inside of the cell, all of this is called the cytoplasm. The liquid gooey stuff is called the cytosol. Glycolysis occurs there in the liquid gooey cytosol. Okay? Then we have the transition or intermediate stage, and we enter into the mitochondria with the products from glycolysis. We enter those into the mitochondria, into the citric acid cycle, which is the Krebs cycle, which is the TCA cycle, I told you. And from there, the electron transport system. So all of this happens in the mitochondria. Glycolysis is in the cytosol. So we're going to take this step by step, guys. And trust me, again, we're not going to cover everything, okay? So we'll start with glucose oxidation. So glucose enters the cell through facilitated diffusion, and then it has to get converted to glucose 6-phosphate after it gets inside the cell. This prevents that glucose from being able to go back out of the cell across the cell membrane. But it also allows the cell to take in even more glucose. So we use a small amount of ATP during this process. Now, the whole step of glucose oxidation, or the whole process, is glycolysis, transition, also called the intermediate stage, Krebs cycle, citric acid cycle, TCA, again, choose one, and then the electron transport system. So glycolysis, again, occurs in the cytoplasm of the cell. We bring that glucose in, and remember I said we had to use a little bit of energy? It takes 2 ATPs to convert glucose to glucose 6-phosphate. Okay? So far so good? Now, we started out spending money. We hadn't made any yet. It's sort of like you have to go buy new clothes to go to your job. You spend money before you make money. So we're spending 2 ATPs to have the energy to convert glucose. Okay? The next thing that happens, oh, sorry about that, guys. The next thing that happens is we're going to start getting ready to make some ATP. So that first part of the image we just looked at right up here, that's actually 5 steps required to make that happen. Now before we go any further, you'll notice the screen suddenly has some drawings on it. I paused for a moment, but the only thing I could find to draw on the screen with was the stylus for my phone, and it really doesn't work well with my computer, so this is terrible, guys. But let me walk you through what I've done here. Glucose is a hexagon-shaped molecule with 6 carbons. So instead of having a hexagon, I just drew circles to indicate those 6 carbons in glucose. So when we use this ATP to get the process started, we break glucose not into what we have already, but we break this bond right here, and that gives us 2 chains of 3. Okay, so there's 3 glucose, here's 3 carbons, here's 3 carbons, okay? In addition to that, those phosphates that we took off your ATPs and turned them into ADPs, we took those phosphates and hooked them on those carbons, okay? Now, we're at this point, we have broken glucose down a little bit, but we still got to keep working. So we're going to continue to add enzymes to this reaction, or enzymes are still a part of this reaction, and now we're going to continue breaking this down. All right, still with me? Okay, let's keep going. Now when we're breaking down glucose, we're changing its chemical structure. We're breaking chemical bonds, but we added phosphates back to it. So the next thing we're going to do is actually add a couple more phosphates that were just sort of, we're going to add a couple of more phosphates that were just there in the cell. So imagine we put another phosphate here and another phosphate here, okay? Now the enzymes will continue to work on this, and we're going to take some ADP and steal these phosphates. So we're going to take this phosphate off and we're going to hook it on here and make an ATP. Take this phosphate off, hook it on here, and make another ATP. Okay? So we're doing that to this molecule because remember it had a phosphate on each end and this one had a phosphate on each end. So we're going to make 4 ATPs by taking these phosphates off and hooking them on the ADP. That is substrate level phosphorylation. We're adding phosphates onto the substrate. Okay? Now, when we do all that, we end up with 2 molecules of something called pyruvate. Now, as we were breaking down glucose, we weren't just breaking it into smaller pieces and adding phosphates. We were also working with our NAD, because remember this started out as NAD+. So remember that plus tells you it's lost electrons, or it lost 1 electron. So as we're breaking down these molecules, we're also pulling off electrons and hydrogen ions. The NAD is going to take those hydrogen ions. Remember, hydrogen ions are positively charged. And what happens? We end up, instead of 2 NADs, now we've converted that to 2 NADH molecules. So I like to think of our NAD and the other one we'll see in a minute, FAD, as being like suitcases. So what do you do with your suitcase if you're going on a trip? You pack it, but you don't unpack it when you're at the airport. You don't unpack it while you're on the plane. You unpack it when you get to the final destination and you're ready to use the things in your suitcase. So if you think of NAD and FAD as suitcases, you're packing electrons and hydrogen ions in there during this process of breaking down glucose. Okay? We're going to take them all the way to the end and unpack them and use them. Now, in addition to packing our suitcases and making some ATP, we also created a little bit of a waste product called CO2, carbon dioxide. Now, let's think about what we've done here. In glycolysis, we spent 2 ATP, but we made 4, so that gives us a gain of 2 ATPs. We packed our suitcases with electrons and hydrogen ions, and we made some trash, CO2. Now that brings us to aerobic versus anaerobic respiration. Now remember guys, respiration in this context does not mean breathing. This is making ATP, okay? So here on the left side of the screen, we've got our glucose, gets into the cell, through glycolysis, we make some ATP, we pack our suitcases, and glucose has now been converted to pyruvate. When there's plenty of oxygen present, the pyruvate will get transitioned into another molecule that enters the mitochondria, gets into the Krebs cycle, and the pathway continues. Now we'll pause right there for this side. Let's go over here to anaerobic respiration. Anaerobic means low or without oxygen. So oxygen is the determining factor here of what happens at the end of glycolysis. So we start out with the same thing. Glucose gets into the cell, we make a little ATP, we pack our suitcase, we end up with pyruvate, but if there's no oxygen present, we can't get into the mitochondria. So this pyruvate is going to be converted by lactic acid. So the NAD will transfer will give those hydrogen ions back to the pyruvate. That transforms it into lactate, which is lactic acid. Lactic acid fermentation will make a few ATP, but it causes muscle fatigue. Now, when we have prolonged problems and we don't have enough oxygen, the anaerobic metabolism can put our body pH out of whack. Now, before we move on to the next slide, where we're going to pick up with the Krebs cycle, I want to use this slide because of the image we have here to talk about how pyruvate gets changed to enter into the Krebs cycle or the citric acid cycle, which is inside the mitochondria. So right here, before that pyruvate does anything else, it starts entering into the mitochondria, we have what is called the intermediate stage or transition. So there's an enzyme that brings the pyruvate together with coenzyme A that's already present. It forms something called acetyl-CoA that then enters the citric acid cycle. So we have to convert or transform pyruvate to acetyl-CoA. Acetyl-CoA enters the Krebs cycle. Okay, so as I said, the remaining stages are aerobic. They require oxygen and they occur in the mitochondria. So let's look just a little bit about this mitochondria since we haven't done the cellular organelles yet. So notice that it is a double membrane organelle. So you've got the outer membrane and the inner membrane. The inner membrane has folds that are called cristae, and the space between the two membranes, what you see here, the yellow and the red, is the outer compartment. The inside part is called the matrix. So the intermediate stage, that transition and the Krebs cycle occur in the matrix. So the molecules of the electron transport system that we're going to see are embedded in the cristae. Remember, the cristae is the inner folds. And this, of course, just repeats what I've already said. Guys, I've already warned you. If there's a picture, I'm going to talk about it. So obviously I got ahead of myself. But the intermediate stage, again, as I mentioned, is sometimes called transition. And it links glycolysis with the Krebs cycle. So that pyruvate is combined with the coenzyme A, forms acetyl-CoA. Energy is released as we pack our suitcase. It occurs twice because two NAD molecules are produced from that original glucose molecule. Two pyruvates are produced, and the acetyl-CoA then enters the citric acid cycle. All right, so let's move on to the citric acid cycle, Krebs cycle, TCA cycle. Again, you'll see I use different terms, but remember, guys, it's all the same thing. So during the Krebs cycle, the acetyl-CoA that we produce during transition is converted to two molecules of carbon dioxide. a CoA molecule is released, energy is transferred to form one ATP molecule. We pack some more in our NAD suitcase. We pack some FAD suitcases. So we create a total of 3 NADHs and 1 FADH per molecule of acetyl-CoA that entered. Okay, remember when we broke down glucose, we made 2 pyruvates. We converted those 2 pyruvates to acetyl-CoA. So 2 acetyl-CoAs came into the mitochondria. So when this says 2 turns for 1 glucose, that's from our 2 acetyl-CoA molecules. So we make a total of, in the Krebs cycle, 2 ATPs. We pack 6 more in our suitcase and 6 more in our FAD suitcase. In addition to that, we also create more CO2. So remember, CO2 is the waste product. When we exhale, we get rid of it. This slide simply gives you a graphic representation of everything we've done so far with cell respiration, where it occurs. Does it use oxygen? What are we working on? That's the substrate. What's created? That's the product. What happens? Is it a pathway or do we build a complex of something? What do we make? And how does oxygen affect it? Have you noticed yet we haven't made a whole lot of ATP? Yeah, so we're about to change that. So in the first 3 steps, we have completely broken down glucose. So what's left? What's the function of this electron transport system if we've already broken glucose down? Well, the electron transport system involves the transfer of electrons, energy, from the coenzymes NADH and FADH2. Now, we produce those during the first 3 stages of cell respiration, and the energy released from those coenzymes is going to be used to form ATP. So the electron transport system is where most of the majority of the energy that was originally in the glucose molecule is transferred to the coenzymes, or was transferred to the coenzymes, and now it's going to be released from those coenzymes and transferred to form our high energy bond that attaches that other phosphate to the ADP, and ATP is made. So the function of the electron transport system, well, look at the name of it, transport electrons. So it transfers electrons from our carriers, the suitcase, and uses that energy to make ATP. So the electron transport system is a series of proteins embedded in the inner membrane, the cristae of the mitochondria. They work as proton pumps. So you can see here our NADH plus the hydrogen ion. We take that hydrogen ion and we pump it out, and that leaves us with the NAD plus. We do the same thing with FAD. We unpack everything, pump the electrons out, and we're left with FAD. Now, this is going to create a gradient between the outer part of the mitochondrial membrane and the inner part. Electron carriers, which are part of this pathway as well, transport electrons between the hydrogen pumps. So this series of pumps and electrons is the electron transport system. Now, a couple of important things here. In order for the electron transport system to do its job, there has to be plenty of oxygen available. So it is an aerobic process. It's oxidative phosphorylation. Remember, we covered that already. It occurs in the mitochondrial cristae, which is the inner membrane. It's a slow process, but it makes the most ATP. So if you look right here, the electrons are transferred from NADH and FADH2 through that series of carriers in the electron transport chain. Oxygen accepts the electrons, okay? So the oxygen, electrons, and hydrogen ions join to form water. But without sufficient oxygen, we can't unpack the suitcases. All right, let's look at this a little more with a little more detail, but still not as much as we could. So the electrons are transferred from the coenzymes to oxygen. So the coenzymes, remember that's our NADH and FADH2, release the hydrogen ions and the electrons, and it becomes oxidized. These electrons are passed through the electron transport chain to oxygen, which serves as that electron acceptor. The oxygen then, combined with those electrons and hydrogen ions, produces 2 molecules of water. So oxygen is a reactant in the electron transport system of cell respiration, and the oxygen becomes part of the water that's produced. So the oxygen you breathe is converted to water within the mitochondria of your cells. Now, converted, maybe. It's used to make oxygen. Step number 2 establishes the hydrogen ion gradient. So as electrons are falling and pass through the electron transport chain, their kinetic energy is harnessed by those hydrogen pumps, and they move hydrogen ions from the mitochondrial matrix into that outer compartment. That creates a gradient with more hydrogen ions outside the cristae. Then that hydrogen ion gradient is harnessed to form ATP. So hydrogen ions move down the concentration gradient as we transport it across the inner membrane by ATP synthase, which is an enzyme. It moves from the outer compartment into the matrix. This process is sort of like water falling over the dam and turning the water wheel. The kinetic energy of the falling hydrogen ions is harnessed by the enzyme to form a new bond between existing ADP and phosphates, hooking those phosphates onto the ADP, and now we have produced ATP. This image gives you a graphical representation of what we just talked about, of the electron transport chain. So now let's look at our totals. What have we made so far? Well, in glycolysis, we gained 2 ATPs, and we packed our suitcase, and we're going to use those later to make more ATP. In the intermediate stage, well, that was our transition. We didn't really make anything. We packed some more suitcases we're going to use to make ATP. In the Krebs cycle, we made some ATP. We packed some more suitcases that we're going to use to make more ATP. So what have we done here? We have created a total of around 32 to 36 ATP. Now, because we used some, we lost 2 right off the bat, and depending on the cell and where that cell is located, you may see anywhere between 30 to 36 as the number quoted. Now, I was always taught somewhere between 30 to 36 ATP, but your book insists on using 30, so guys, we're going with 30. But that's a lot of ATP. But when you think about how much our body uses, that's a drop in the bucket. It's basically like taking an eyedropper of water and adding it to the ocean. Cyanide is formed when a nitrogen atom triple bonds with a carbon atom. Cyanides are extremely toxic if they combine with a specific carrier in the electron transport chain. That leads to inhibiting the electron transport chain and therefore leads to inhibiting the production of ATP. Even though we've got plenty of oxygen available for catching the electrons with the electron transport chain, when you inhibit that one electron carrier, it prevents the electrons from reaching the oxygen. So the aerobic components of cell respiration progressively just decrease as the cells can't use the oxygen and leads to cyanide poisoning, which is most harmful to the organs that require the most oxygen, such as your heart, your lungs, and of course the brain. Treatment for non-lethal cyanide doses, when it's just a little bit, involve administering substances that will just bind to the cyanide and then help them to be eliminated in the urine. At the beginning of this discussion, I mentioned that we can use other things such as fatty acids and amino acids to oxidize them and make ATP. Fatty acids are going to be enzymatically changed to 2 carbon units at a time to form acetyl-CoA. We call that beta oxidation. The acetyl-CoA then enters the pathway at the Krebs cycle. Now, this can only be oxidized aerobically, so oxygen must be present. One of the things that happens because of this is the production of keto acids, which can be very harmful if the keto acids accumulate, as they do with uncontrolled diabetes mellitus, and the patient goes into ketoacidosis. Amino acids use a different pathway, but the pathway depends on what the specific amino acid is. So there are different amino acids that enter the pathway at glycolysis, the intermediate stage, or directly into the Krebs cycle. The amine process of amino acid is a waste product that's going to be converted to urea and excreted by the kidneys. Now, what's missing in both of these pathways? Any ideas? Well, for the most part, it's glycolysis. There are a few amino acids that can enter glycolysis, but it is very rare. So fewer ATP molecules are created. All right, congratulations, guys. You made it through cell respiration.